Assalamualaikum warahmatullahi wabarakatuh. Today, my group and I will be presenting about the application of recombinant DNA. Recombinant DNA is a DNA molecules that are produced by two different species which are inserted into a host to form a new genetic combinations. For this presentation, we'll be looking at the use of recombinant DNA in the medicine aspect, which is the gene therapy of cystic fibrosis. Cystic fibrosis is a genetic disease which causes lung infection and may cause difficulties breathing in a long term. This occurs due to the mutation in the cystic fibrosis transmembrane conductance regulator, which causes it to become dysfunctional.
Gene therapy utilizes the delivery of DNA into cells, which can be accomplished by several methods known as recombinant virus, the viral vectors, and DNA complexes, the non-viral vectors. In gene therapy of cystic fibrosis, the viral vectors method are used to treat the disease. The vector used in this therapy is known as lentiviral vector. Lentiviral vectors in gene therapy is a method by which genes can be inserted, modified, or deleted in organisms using lentivirus. Lentivirus is a retrovirus, meaning it has a single-stranded RNA genome with a reverse transcriptase enzyme. Lentivirus will be delivered a healthy copy of a gene called CFTR, which causes cystic fibrosis if it carries a mutation, unlike other retroviruses, which cannot penetrate the nuclear envelope and can therefore only act on cells while they are undergoing mitosis. Lentivirus can infect cells whether or not they are dividing. Scientists use the lentivirus mechanisms of infection to achieve a desired outcome to gene therapy.
Integrating gene therapy is where a piece of DNA that contains a correct version of CFTR gene will be delivered to an individual cells and become a permanent part of their genome. The patient will receive the gene therapy only once or a few times in his life. An integrating gene therapy to treat cystic fibrosis is being tested in animals, and a clinical trial to test the safety of this therapy in people with cystic fibrosis could happen in the next several years. Next, non-integrating gene therapy. The procedure of this gene therapy is same with integrating gene therapy, but the CFTR gene remains separate from the genome. The cell can still use the new copy of the CFTR gene to make normal CFTR protein. The effect of the gene therapy might last only for several weeks or months. Non-integrating gene therapy is the safest type of gene therapy for cystic fibrosis patients. In a clinical trial in England, people with cystic fibrosis were given a dose of a non-integrating gene therapy once a month for a year. The result of the gene therapy was safe and resulted in a small improvement in lung function. RNA therapy is where an mRNA is inserted directly into the cell and the sequence is used to make functional CFTR protein. The patient will receive the treatment several times. A potential RNA therapy for cystic fibrosis is being tested in early stage clinical trial to determine its safety.
Upon research, non-integrating gene therapy for cystic fibrosis was chosen as it is safe and successfully showed a small improvement in lung function. This was proven in a clinical trial in England where people with CF were given a dose of a non-integrating gene therapy once per month for a year. Nevertheless, additional work is needed to improve this kind of gene therapy so that it will be more effective at treating cystic fibrosis. This method of non-integrating gene therapy here uses liposomes to deliver gene therapy in cystic fibrosis. Patients with cystic fibrosis lack functional cystic fibrosis transmembrane regulator or CFTR protein, which is needed to make thin, watery mucus that keeps the lungs healthy. Normal CFTR gene can be delivered via inhalation of nebulized liposomes mixed with plasmid DNA encoding the protein. The liposomes enhance transport of the CFTR gene into host airway cells via endocytosis or direct fusion with the cell membrane. The plasmid then passes into the cell nucleus where the normal CFTR is transcribed, enabling the cell to make CFTR proteins.
gene therapy has been ongoing for decades now. There is light at the end of the tunnel as the gene therapy is being introduced. As the proof of the gene therapy is that the effect is long-lasting and also timeless, where replacing the defective gene with a functional gene in the disease, it also has limited chances of remission and also will grant you symptom-free for life. The second one is gene therapy can also eradicate diseases and improve the quality of life, where the alteration of gene, especially using germline method, can help avoid the transmission of defective genes and also no further incidence of diseases. The third one is that it is a new source of hope, where the promised cure for chronic illnesses can currently curable. As for the cons of the gene therapy, it concerns that carefully added into gene therapy, as the cost of the performing the gene therapy is likely high because it uses high level expertise. The second one is it has potential of resistant diseases, which is it is unpredictable and also will cause to the worsen situation. The last one is it is an advancement of gene therapy, which will create ethical concerns to the human race.